So the immune system has a specific portion called the adaptive part of the immune system. It has two main parts. The first part is called cell-mediated immunity. This is mediated by specific cells called T lymphocytes. The other part of the specific immune system is called antibody-mediated immunity, sometimes called the humoral immune system, the humoral immune system. This is the part that has antibodies. So first, let's focus on the T cells and the cell-mediated part of the immune system. Both, really, the T cell and the antibody portion of the specific immunity respond to specific molecules called antigens. An antigen is generally a complex molecule, so simple little things like sugars generally aren't antigenic. They won't trigger a specific immune response. But larger proteins, large polysaccharide complexes, even lipids, glycolipids, found on the surface of membranes can serve as antigens. Usually, any large molecule has multiple little spots on the outside of it that could be recognized specifically as foreign by your immune system. Each one of those spaces is considered to be an antigenic determinant. So you could have a molecule that has 10, 15, 20 different spots on the outside of that molecule that could be recognized or could be antigenic determinants. Sometimes even small molecules that are not in themselves big enough to be antigens can actually bind to proteins and become antigenic. Those are called haptens. So hapten is something that's too small, not complex enough to become an antigen itself, but when it binds to something else, it gets big enough to create an antigen. So here's a protein, and this picture is showing you that different T cells each could have a receptor that recognizes a different part on this protein. So in this case, it's showing you 3 different antigenic determinant portions on this protein, and each of those are spots where a T cell could specifically recognize that antigen and create an immune response in response to it. In general, what happens is most of the cells in your body have a cell surface protein called an MHC, or major histocompatibility complex. The kind of MHC found on just about all cells are called class 1 MHC proteins. So what happens is something, a virus or a bacteria, gets inside that cell, and it can actually, it probably will get digested up into little pieces in the cytoplasm of the cell. It can actually bind to the MHC in the endoplasmic reticulum when it's being made, and then when the MHC becomes part of the membrane of the protein, it ends up as a receptor kind of on the outside of whatever cell this is. It could be any cell, and it's got a piece of the virus or a piece of the bacteria that it's kind of holding out on the outside of the cell. And in effect, antigen presentation is any cell has got its MHC and it's holding out this piece of a virus or a piece of a bacteria to the T cells going, hey, do you see this? Do you see this? Is this something that's bad? Is this something that's foreign? Attack it if it is, right? So imagine all the cells in your body, if they are invaded by anything, have this capability of taking pieces of that thing and holding it out and going, hey, come, get rid of this thing for me if your body has any T cells that are capable of recognizing it. Professional phagocytic cells, like the macrophages and the neutrophils, when they phagocytose something, they're like engulfing things that are outside the cell, bringing them in to lysosomes, breaking them up into pieces, and they have another kind of MHC called class 2 MHC. So the antigen presenting cells, the phagocytes, have this special MHC. They really spend a lot of time getting all the pieces of things they phagocytose and waving them on their cell surface, going, hey, check this out. Is this a bad thing? So how do the T cells get this specific immunity? How do they know what it is they're supposed to attack? Well, T cells begin as lymphocytes in the bone marrow, so the stem cell that's capable of making red blood cells, capable of making macrophages for that matter, makes lymphocytes. A special kind of lymphocyte is a T lymphocyte. This crazy process happens in the T cell when it gets made. There's a large region of the DNA that gets alternatively spliced and makes a receptor. So every T cell in its development randomly gets their DNA spliced such that they make one kind of receptor. And that receptor is the thing that's going to be able to recognize an antigen. Okay? So imagine a T cell gets born in the blood, and in the process of its birth, it makes one kind of receptor. That receptor goes on the surface of this cell, and then T cells have to go to school. So T cells are released from the bone marrow, and they go to a special organ called the thymus. So the thymus is a special organ, to our knowledge. Its major purpose is as a school for T cells. So the T cells go in, and the first part of their schooling is, their first major test is called positive selection. So in positive selection, they look at the T cells and see if their receptor that they made can bind to MHC. If their receptor can bind to MHC, they pass that test, they're able to graduate, and they go on to the next test. If their particular receptor can't bind to MHC, they flunk the test, they're killed. Done, dead, can't go on. This is a brutal school. So all of the T cells that have defective receptors that can't recognize MHC, they're not ever going to be able to do their job and recognize antigens presented. They're killed off. So the ones that survive positive selection, we know they have MHC on their cell surface. Well, they have receptors on their cell surface that can bind to MHC if they need to. So then they go on, their next major test is called negative selection. So these T cells now have their receptors, and they basically in the thymus go through, and their test is they're presented with a bunch of MHC that have antigens that are normal antigens that should be found in your body. If that T cell ever recognizes, binds to an MHC that has a normal protein that should be found in your body, it's killed. Because it would be really bad if in fact a T cell went out and started killing off normal things that should be found in your body. That's really a bad thing. So negative selection is they can't kill off things that should normally be found in your body. They should only be looking for things capable of finding things that shouldn't normally be found in your body. Okay, so if they pass negative selection, they are now capable of binding to a cell that has MHC on its cell surface, and they're not going to bind to something that's normally found in your body. They graduate. They get released from the thymus school, go out to circulate through your blood and through your body. They are called immature thymocytes, frequently called virgins. So virgins are cells that have been released from school, but they haven't ever found the antigen they were made against. So they're released. Most of them spend their lives going through your body, and they never find that one special cell that has the antigen they were made against, and they die. However, if in fact this virgin cell goes out and someday finds a cell that has MHC and has the antigen that just by luck this T cell receptor happened to get put together by the random splicing of DNA, and it happens to match that particular antigen, it now binds to the antigen it was made against, and now it clonally expands. Now, instead of one T cell, it's found the antigen it was made against, it makes thousands of T cells. Most of the T cells that get made from that one activated, no longer a virgin T cell, actually become killer cells. Some of them will actually become memory cells. Once a T cell finds the antigen it was made against, some of the cells get to be memory cells, and those memory cells are capable of remembering that antigen for many, many years. So once it's been activated, now if you ever run into that particular antigen again, you've got T cells all over your body that remember that specific antigen and are ready to attack it again. At the same time, tons of activated killer cells are made that go out and kill any more of those antigens that happen to come into the body. So you also make a special kind of T cells. So there's multiple different kinds of T cells. You have the killer ones, also called cytotoxic T cells. What does cytotoxic mean? Toxic to cells or killers. So they will basically release enzymes that will completely cause a cell to rupture. You can also have helper T cells. Helper T cells are pretty amazing cells. Once a helper T cell finds the antigen it was made against, it not only activates more T cells, it can activate the humoral part of the immune system, the B cells, and it can activate the non-specific part of the immune system as well, inflammation, all of those different parts. So helper T cells are getting the whole immune system involved in fighting this, whatever it is. So here's a picture of a T cell, and it's come upon a cancer cell. So this T cell has a receptor that recognizes MHC on this cancer cell, and whatever the antigen is that that MHC has, the T cell can recognize that it is the antigen it was made against that is not right, not supposed to be in your body, and it attacks it and basically causes the cancer cell to explode. This, you can see like blebbing parts of the cell membrane just falling off the cell. So here's showing you the helper T cells release factors that trigger macrophages and inflammation. It releases factors that cause B cells to start expanding, and it triggers more T cells to get in there and kill things. So helper T cells are kind of like the generals in the army getting everybody involved in the battle. So from the activated T cell that has found its antigen it's made against, you now have cytotoxic T cells, the killer cells that go out and explode cells directly. You have memory T cells. They're the ones that stick around and remember for years, 10, 20, 30, maybe your whole life, that specific antigen and are ready to attack it. There are also types of T cells called suppressor T cells. Because you can imagine, if every T cell just activates more and activates more, pretty much everything is going to get killed off. Your inflammation gets started. It would be a bad situation. So suppressor T cells are actually in there to help kind of moderate it, help slow it down. So in fact, hopefully, the T cells only attack the specific antigens they were made against, and if not very many antigens of those are around, the T cell response can kind of get shut off and it'll stop. You have helper T cells as well, which are the generals that I told you about that are bringing everybody into the battle.